The nervous system. It is the most complex of the body's systems, but it has only 5 major kinds of components: brain, spinal cord, nerves, ganglia, and sensory receptors. It generates and transmits electrochemical phenomenon called nervous impulses and uses these to stimulate muscle tissue and glandular epithelium. Although the heart is often viewed as being the emotional center and soul of an individual, physiologists have established that the essence of our being lies within our brain. When someone receives a heart transplant, they are the same individual with a different cardiovascular pump. But if medical scientists are ever able to transplant a human brain, the recipient will be who the donor was.
Hi, I'm Mike Shively. Welcome to Introduction to Anatomy and Physiology. Today we want to talk about the nervous system. The nervous system is the most important control system of the body, and it exerts its control over other parts of the body by way of electrochemical phenomena that we call nervous impulses. Now there's another system in the body that exerts some control over the body. What system would that be? Eva?
The endocrine system?
The endocrine system. The endocrine system exerts its control by chemical phenomena that we call hormones and chemical regulators. But the nervous system generates nervous impulses and interprets those nervous impulses. Let's go to the overhead camera for a minute. I want to put up some acronyms that most of us are probably familiar with already. When we say CNS, what comes to mind, somebody? Adam?
Central nervous system.
Central nervous system. And there are only 2 organs that are included in the central nervous system: the brain and the spinal cord. PNS is another common acronym that we use in the nervous system. What does that stand for? Starley?
Peripheral nervous system?
Peripheral nervous system. Oh, we've already got it down there, PNS. What I want to do is put what is included. Nerves, ganglia, and sensory receptors. Finally, we often see this acronym for part of the nervous system. ANS stands for autonomic nervous system, and it's actually not, I'm going to put parentheses around it because it's not a physical subdivision in the same sense that these two are. It is in fact a functional subdivision, and it's the part of the nervous system that sends motor impulses out to what we call involuntary effectors. Who can tell us what the involuntary effectors are in the body? Adam?
Cardiac muscle tissue is one of them.
Cardiac muscle tissue is one. Another one? Hazel?
Smooth muscle tissue.
Smooth muscle tissue. And the third one is, Eva?
Glandular epithelium?
Glandular epithelium. So this is a part of the nervous system that actually is a functional subdivision rather than a physical entity in and of itself. So once we've said CNS and PNS, we've really included all the components of the nervous system under those 2 umbrellas, if you will. So we really only have 5 kinds of organs: brain, spinal cord, nerves, ganglia, and sensory receptors. Certainly there's a lot of subdivisions of each of these. One can take the nerves, for example, and divide them into cranial nerves and spinal nerves. The brain itself is the most complex organ anatomically in the body. In fact, if we consider that there are about 10,000 parts of the body that are large enough to be seen and named, seen with the naked eye and named, about 1,000 of those, or about 1/10 of the gross anatomical structures, are actually brain subparts. So it's a very complex organ. Eva?
I just have a question about one of the terms you used. What are ganglia?
Good question. What are ganglia? Let's go to the overhead camera and put that word back up so we can define it for everyone. Singular form of that word is ganglion, or ganglia is plural. These are neuron cell bodies in the peripheral nervous system. So we have groups or collections of neuron cell bodies that are outside of the brain or spinal cord. They're in the peripheral nervous system. The term for a singular version of that is ganglion, or the plural version would be G-L-I-A, ganglia. Now, there's a parallel term for groups of neuron cell bodies that are found within the central nervous system. Within the brain or the spinal cord, we also obviously have groups or collections of neuron cell bodies, and each one of those is a nucleus. Same word that we used for the central component in a cell that contains the DNA. So these are neuron cell bodies, a collection of those, a group, if you will. In the CNS. Again, I didn't put it down, but this has to be a group of these and a group of these. So the only difference basically between a nucleus and a ganglion then in this usage is location. It's location. While we're on that, a couple other parallel terms that we ought to define as well. Let me just draw a line here, and let's write the word nerve. Probably ought to define what that is, since this is a beginning session on the nervous system. And that is actually a collection of axons and or dendrites. These are processes of neurons within the peripheral nervous system. So we'll just put down a bundle of axons or dendrites in the PNS. PNS. With the same concept, though, within the central nervous system, just like we had a difference up here, depending on where we were, if we have a collection of neuron cell body, or axons or dendrites, rather, in the peripheral, central nervous system, we call that a tract or a fiber tract. So this is basically the same thing in the CNS. So the cell bodies are ganglia or nuclei. Their axon or dendritic bundles are either nerves or tracts, depending on where we are. Any other questions while we're getting some basic definitions and concepts out of the way? Starley?
Are there no axons or dendrites that actually move from the PNS to the CNS, like actually cross that border?
Well, there are axons and dendrites, Starley, don't move within our body, but obviously there can be some neuron cell bodies in the CNS that send axons out into the peripheral nervous system. So they cross that border. They don't physically move, but they extend out into the peripheral nervous system. Does that make sense? Mm-hmm. Let's take a look at a graphic that summarizes the subdivisions that we talked about on the nervous system. We've got these three subdivisions then: central nervous system with the brain and the spinal cord, peripheral nervous system with nerves, ganglia, and sensory receptors, and then this one down here that we said we probably ought to offset or think about a little bit separately, because it actually includes some parts of the CNS and some parts of the PNS. It's not an anatomic subdivision, but rather a functional subdivision. But it has enough peculiarities that we we'll consider it separately in a later session. Now, when we think about what the nervous system does from a functional standpoint, there are really not a whole lot of things. We can kind of group them down into just 3 or 4 kinds of things. And let's separate the central nervous system from the peripheral nervous system when we think of the functions that each of these perform. They're somewhat related, but there are some subtle differences as well. We'll begin with the central nervous system. What does the brain and spinal cord do? What is their function on a daily basis?
It sends out impulses.
They send out impulses, and they send out those impulses really only to 2 kinds of places. When you send out an impulse going out of the brain or spinal cord, we call that impulse a motor impulse. And there's only really 2 kinds of destinations for those. One is some muscle tissue someplace, and the other is some glandular epithelium. So we send out motor impulses to muscle tissue or glandular epithelium. That's one of the things that the brain and spinal cord do. Another one, Eva.
They receive sensory impulses.
Okay, the CNS receives sensory impulses. You know what? Let's put those words up here, motor and sensory. Go with the overhead camera for just a minute. When we say motor, in this context, we're going to be talking about impulses that are leaving the brain and spinal cord. They're going out of the central nervous system. So we'll just say impulses leaving CNS. And when we say sensory, we have the opposite concept. We're talking about impulses that are coming into the CNS. And some other words that we probably ought to be familiar with in this regard are efferent. Efferent is, for our purposes, synonymous with motor, and afferent is synonymous with sensory. All right, we said that the brain and spinal cord send motor impulses out to muscle tissue and glandular epithelium. The brain and spinal cord receives sensory impulses in from the peripheral nervous system. And one other thing that the brain in particular does. Seth?
The place where we carry out conscious thought and store memories and
Thought,
things like that.
memory, and interpretation of those sensory impulses, you bet. In other words, the processing of information. Sometimes our brain doesn't process as well as we'd like. Sometimes it doesn't store as well as we'd like. Sometimes it misinterprets. Nonetheless, we have these three general kinds of functions for the central nervous system. Send motor impulses, receive sensory impulses, and store and process information. The peripheral nervous system has somewhat parallel functions. I think we can probably figure out what those are. If the brain and spinal cord are going to send motor impulses out to muscle tissue and glandular epithelium, then we're going to have some nerves within the peripheral nervous system that are going to carry those motor impulses. So the peripheral nervous system distributes the motor impulses out to the various effector organs. It distributes those motor impulses. They originate in the brain and spinal cord, but they're carried on out to their destinations within the nerves of the peripheral nervous system. And by the same token, we said the CNS receives sensory information. Well, that sensory information is actually originally generated out in the peripheral nervous system by sensory impulses, or by sensory receptors, rather. And then sensory neurons carry those impulses in. So if we'd kind of say that there's three functions of the PNS, we'd say carry motor impulses out to the effectors, generate sensory impulses, and thirdly, carry those sensory impulses into the brain or spinal cord. Adam?
Are there physical anatomical differences between sensory neurons and motor neurons?
We're going to talk about neurons in particular here in just a little bit, and we'll actually classify neurons into three physical groups, if you will, and four functional groups. But it is intriguing that an impulse in and of itself is really no different whether it's a sensory impulse coming into the brain or spinal cord or a motor impulse going out. It's the same electrochemical phenomenon. But it is carried by different neurons, and that's one of the things that a lot of people are surprised about. They kind of think maybe a neuron could carry a motor impulse now and a sensory impulse in a moment, and that's simply not the case. Neurons are one-way streets, really, physiologically for impulses. So a given neuron either carries motor impulses out to a certain place or it carries sensory impulses in from a certain place. but never does both at the same time. Starley?
I know that you said within the CNS that it can only go out to skeletal muscle tissue or involuntary or glandular epithelium, something of that nature. Is the sensory receptors the same? Where the PNS creates them, are they only in those same skeletal muscle tissue and the involuntaries, or can they be
Well, we've got
elsewhere?
sensory receptors all over our body, to be honest with you, and there are a lot of different kinds of them. Their job is to generate impulses, and then those impulses are carried in to the brain and spinal cord, where those impulses are interpreted as hot, cold, pressure, pain, touch, or vision, hearing, equilibrium, and those sorts of things. So I'm not sure, your question was what again now?
Oh, I didn't know if where they were sending the impulses to or where they were coming back from, like where those sensory receptors are. But obviously you were saying the sensory receptors are in a lot greater areas than where they're sending the motor impulses to.
Well, a given sensory receptor, that particular sensory receptor synapses with a particular sensory neuron that carries the impulse that the receptor generates on into the brain or the spinal cord. So if we're talking about a receptor that responds to touch or hot or cold out here in my fingertip, those are different sensory receptors. Every one of them has a different sensory neuron that carries the impulse in.
Okay.
And when the impulse gets into the brain or spinal cord, all the impulses are the same. There's no difference between an impulse that's going to be interpreted as heat versus an impulse that's going to be interpreted as cold, or an impulse that's going to be interpreted as touch or pressure. It's the brain interpreting those electrochemical phenomena from a certain location that determines what we feel as heat or cold or pain or pressure.
Thank you.
Sure. Let's go to a graphic that summarizes the functions of the CNS. I don't believe we have looked at this graphic yet. Receives sensory impulses, sends motor impulses, and processes and stores information. And we'll go to the next graphic because we've already talked about many of these things already. Basic definitions involving the nervous system. Again, when we say motor or efferent, we're talking about impulses going out of the brain or spinal cord. Now, we can apply these adjectives to both neurons or the impulses that they carry. So if we say a motor neuron or a motor impulse, the idea is the same. We're talking about impulses or neurons that are carrying those impulses out of the brain or spinal cord. Sensory or afferent impulses or neurons are implying the impulses are coming into the brain or spinal cord. A nucleus we define as a group of neuron cell bodies within the CNS, and a ganglion, same concept, but out in the peripheral nervous system. A fiber tract was a group of axons or dendrites, a bundle of them, if you will, within the central nervous system, and a nerve was the same concept in the peripheral nervous system. And then we've used this term neuron already. Let's all remember that when we say neuron, we're talking about a nerve cell. Let's go ahead and talk about neurons for just a little bit, since they are the functional cells of the nervous system. There are actually two different groups of cells within the nervous system. The ones that transmit the impulses, neurons, and the ones that support the neurons in various ways. And those cells are called glial cells or neuroglia, and we'll consider those separately here in a bit. Let's go to the overhead camera and we'll make a schematic representation of a neuron. Some neurons have a cell body. These are going to be extremely schematic. This will be the nucleus within it. And they have a single process sticking out either side. If during impulse transmission the impulse goes this direction under physiological circumstances, by definition, this process sticking out this side is called a dendrite. And this process over here is called an axon. We have a few neurons that are actually this shape within our body. And these kinds of neurons with a single dendrite and a single axon and a cell body just right in between the two, these are called bipolar neurons. They're relatively uncommon. Just a few of the sensory neurons have this conformation. Another type of neuron is almost the same, but the cell body is offset. And this, if the impulse transmission is this direction, this would be dendrite, this would be axon, and this particular configuration of a neuron is called pseudo-unipolar. And most of the sensory neurons that we have in our body are this type, pseudo-unipolar. Most sensory neurons. Finally, the last possibility that exists, actually, is we have the cell body and we have multiple dendrites coming in, several different dendrites coming in, and a single axonic process leaving the cell body. And this one is called multipolar. All motor neurons are multipolar. So 3 kinds here: bipolar, pseudo-unipolar, and multipolar. And these are very rare, just a few of the sensory neurons. These are most of the sensory neurons, and multipolar neurons down here make up all of the motor neurons in the body. So those are, we call these the anatomic or morphologic types of neurons. Any questions there? Eva?
I've actually got 2 questions. First, I just want to clarify the definitions of the processes on the neurons. Are the dendrites the ones taking the impulse to the cell body and then the axons carrying it away?
By definition, an impulse that under physiological conditions transmits the impulse toward the cell body is dendrite, and the one that transmits it beyond the cell body is axon.
Okay. And then your drawing of the multipolar neuron, is there ever any time when there's a multipolar neuron that has more than one axon? Or is it only the dendrites that are multipolar?
Technically, neurons only have a single axonic process, although that axon can branch out toward its end many, many times. And dendrites can do the same thing. Even those, the bipolar and pseudo-unipolar neurons that we drew that had a single dendritic process, those dendrites can branch, but only multipolar neurons have multiple dendrites themselves. Any other questions? Seth?
I was wondering what the length of a single neuron is. Do we have just one that runs from our fingertip to our brain, or are there several synapses in between?
Many of the neurons in our body are among the longest cells we have. Actually, some of them have axons a few feet long, have processes a few feet long. The sensory neurons, for example, that carry sensory impulses from our fingertips, the impulses are generated within sensory receptors right there in the skin of our fingertips, but a single neuron carries that impulse all the way up the superior limb into the spinal cord. So you can think of the length there. Motor neurons carrying impulses out to the skeletal muscles within the hand go all single motor neuron goes all the way down to those particular muscles. Same way in our inferior limbs. So some of them are quite long. Now there are other cells in the body that have larger volume, and there are other cells in the body that are bigger in diameter, certainly, but neurons, some of the neurons are the longest cells in the body. Adam?
So if that neuron's damaged anywhere in its path, does whatever it innervates quit working?
If you sever the axon of it, certainly the impulse cannot be transmitted beyond that point of severance. And now that some of them can regenerate, because just because you sever the axon does not mean that the neuron itself is completely wiped out. But that does interfere with impulse transmission, whether it's sensory information coming in or motor impulses headed out. Let's take a look at a graphic that summarizes those neuron types and make sure we've covered all the bases that we intended to cover on those. Multipolar neurons, many dendrites, one axon, all motor neurons. Bipolar neurons have a single dendrite and one axon, but they only compose a few of the sensory neurons. They're not very common. Pseudo-unipolar neurons, one dendrite, one axon, has an offset cell body that kind of distinguishes it from a bipolar neuron, and most of the sensory neurons are pseudo-unipolar in type. Now those are the three morphologic types of neurons. We call them anatomic types because of their configuration. But their neurons are also classified into 4 groups from a functional standpoint. Let's take a look at that on a graphic here. When we talk about somatic efferent neurons, efferent are obviously motor neurons, these are the neurons that are carrying impulses out to skeletal muscle tissue. And that's all they do, carry impulses to skeletal muscle tissue. When we say visceral efferent neurons, we're talking about neurons that are carrying impulses out to smooth muscle tissue, cardiac muscle tissue, or glandular epithelium. These are the three kinds of involuntary effectors that we mentioned a bit ago. Now, which of the three anatomic types of neurons would both of these be? They're both efferent. Adam?
Multipolar?
They're both multipolar. We said that all motor neurons are multipolar. We said that efferent means the same thing for our purposes as motor. So both of these are multipolar neurons. We're just two different kinds from a functional standpoint, depending on whether they're carrying those impulses out to skeletal muscle tissue or whether they're carrying the impulses to smooth muscle tissue, cardiac muscle tissue, or glandular epithelium. Then we get to the two functional types of afferent neurons, somatic afferent neurons and visceral afferent neurons. These, the somatic afferent neurons, these are often abbreviated, incidentally, SA neurons. You'll see that in some texts and probably in the physiology texts when you take that course in the future. But these are sensory neurons that are carrying impulses from relatively superficial structures in the body like skin, muscles, bones, joints. But they also include those neurons that are carrying sensory impulses that are going to be interpreted as vision, hearing, and equilibrium. So basically we say from skin, muscles, bones, joints, from the retina of the eye, and from the internal ear. Any impulses going into the brain or spinal cord from those kinds of locations. Let me say it one more time. Skin, muscles, bones, joints, retina of the eye, and internal ear. Those impulses are going to be classified as somatic afferent impulses, or the neurons that carry them will be somatic afferent neurons. And then the other category of sensory neurons are visceral afferent neurons, and they carry impulses that originate in deeper body structures, like when you have a stomach ache, you know, your stomach doesn't feel good, you feel nauseous. That's a visceral afferent impulse carried by a visceral afferent neuron. And in addition to the deeper body structures, also included here are impulses that are going to be interpreted as taste or smell, sense of taste or smell. So these are what we call the four functional neuron types, or four functional neuron groups. So we have three morphologic types and four functional types. Now, we said a bit ago that we have neurons that conduct impulses, and then we have another group of cells within the nervous system called glial cells, or neuroglia, that don't carry impulses, but they do support the neurons in various physical and metabolic ways. And there are actually several different kinds of these glial cells. There are four kinds of them in the central nervous system within the brain or spinal cord, and there's actually two kinds of them out in the peripheral nervous system. Let's take a look at a graphic that summarizes these. They're called glial cells or neuroglia. Again, remember, they do not conduct impulses. That's a critical point. Some of them are called neurolymnocytes. Now, an old name for neurolymnocyte is Schwann cell. And what these cells do is actually create ensheathments around the axons of many of the neurons, which actually kind of insulates those axons and actually speeds the rate of conduction of impulses up a bit. Another cell that does basically the same thing, but it's found in a different location, oligodendrocyte. It also creates these myelin ensheathments. The difference between these two cells, besides the fact that they have a different location and name, this one, well, it didn't mention the location, I guess. These guys right here are found in the peripheral nervous system, and these cells are found in the central nervous system. They do the same sort of thing, though. They create insulating ensheathments around axons. The astrocytes are glial cells that actually form a kind of a final filter, if you will, for materials that are coming out of the bloodstream before those materials reach a neuron. Neurons are very sensitive cells. As we'll see in a little bit, they carry a bit of a charge in their cytoplasm compared to the extracellular fluid, so they're kind of irritable in and of themselves. And these astrocytes then, anything that's going to get to a neuron has to go through an astrocyte to make it to the neuron, kind of like a Secret Service man around the president, I guess. The microglia are actually phagocytic cells that are found in the nervous system, and they wander around looking for trouble in the form of bacteria to phagocytize. The ependymal cells are just cells that line some of the spaces within the brain and spinal cord. So they're just lining cells within, the ventricles of the brain and are in direct contact then with cerebrospinal fluid found in those spaces. And finally, the satellite cells down here are supportive cells that are found in ganglia. Now, I said that there were 2 of these that were found in the peripheral nervous system and 4 of them found in the central nervous system. The way you have them listed here, the first one and the last one, obviously, in ganglia down here, those are found in the peripheral nervous system, and the other 4, the second through 5th one in our listing here, are found in the central nervous system. So they perform special functions, but they do not conduct impulses. Bryce?
You said that the microglia phagocyze things. What does phagocyze mean?
Oh, phagocytize. Let's go to overhead for just a second. Phagocytosis is a form of endocytosis, Bryce, which means getting something into a cell. Let's say this is a cell sitting here, and this may be something in the form of a bacterium or something else. In this case, if this were a microglial cell, this cell could actually engulf this bacterium by forming some pseudopods that come out here and surround this guy, and then these meet here and basically encapsulate this foreign object. And so then this particular material would be in the cytoplasm of this cell. So this is one of the ways that it's kind of a protectory system for the nervous system to have these phagocytic cells that have the capability of engulfing foreign material present in the brain, spinal cord. Any other questions on those? Yeah, Starley?
Are there ever ensheathments around dendrites?
Let's see. I don't think that dendrites are ever myelinated. Let me show you. Let's go to the overhead camera a little bit. Let me just show you, since we're talking about this. Here's a model of a multipolar neuron. And here's the cell body, lots of dendrites sticking out here. You can see axonic endings coming in, making synapses with some of these dendrites. And then this represents the axonic process of this particular neuron right here. And you can see that there are layers of cell that have been wrapped around this axon. So this is actually a neural lemmacyte or Schwann cell. Here's another one, and there will be another one on down here. Each one can only cover a small segment of the axon. But by wrapping themselves around and then containing this lipid material in their cytoplasm called myelin, they effectively insulate this axon, and it also speeds impulse transmission. We're going to talk about impulse transmission here in just a second, but when we have myelin ensheathments around an axon, it speeds that impulse transmission tremendously. But your question, I don't believe we have dendrites that are myelinated, just axons. And not all axons of all neurons are myelinated either, Starley. It's just some, actually quite a few in the peripheral nervous system, and some in the central nervous system that are myelinated. Any other questions? Let's talk a little bit about a nervous impulse. Just how does this nervous impulse occur, and what really is it? Let's go to the overhead camera, and we'll draw a schematic representation of a neuron. We'll not worry about how far he goes or anything like that. We'll just say this is a nucleus. And as he sits there at rest, not transmitting an impulse right at the moment, he's got about a -70 millivolt charge in the cytoplasm compared to the charge in the extracellular fluid. Now, a millivolt is only 1,000th of a volt. So this is not going to light up Orem and Provo at night, certainly. But it is a little bit of a difference in charge in the cytoplasm compared to the outside fluid. Out here, there are a lot of sodium ions. Remember, the chemical symbol for sodium is Na, and if it's ionized, it's Na+. So I'm just going to write a bunch of Na pluses out here to symbolize that there's a lot of sodium in this extracellular fluid. There's not very much sodium in the cytoplasm, so I'll put one Na plus in there and put it in parentheses. Yeah, there's some in there, but not very much. What we do have a lot of, ion-wise, in the cytoplasm, though, is potassium, K+. There's a lot of potassium ions in the cytoplasmic fluid. So this is kind of the picture we have of a neuron sitting at rest. Minus 70 millivolts charge with lots of sodiums on the outside and lots of potassiums on the inside. Now, when a neuron is at rest, the cell membrane is not particularly permeable to the simple diffusion of sodium and potassium. Now, you might ask, where does this minus 70 come from if we've got a bunch of positively charged potassium ions in the cytoplasm? And the answer basically to that is that most of the minus 70s from other ions. There's a lot of negatively charged protein ions that are in there, enough that it basically overwhelms the positive charge produced by the potassiums. Now, to get an impulse initiated in this neuron, we have to do something to the cell membrane to bring the voltage up to about minus 40. That particular place that we have to raise the resting voltage to, from minus 70 to plus 40, that particular number is called the threshold potential for that particular neuron. If we can reach that voltage, then it'll transmit an impulse. If we can reach that voltage, it'll transmit an impulse. So let's go back to our overhead for a second, and we're going to assume that right in this location, down here at this end, we do something to the cell membrane right there that raises the voltage up to about minus 40. And if that happens, there are conformational changes in the proteins within the cell membrane of the neuron that allow sodium ions to diffuse into the cytoplasm. Now, I think you can understand why sodium, given the opportunity, would readily diffuse in. There's a negative charge in there, and sodium is positively charged, so we have that electrical gradient. And in addition to that, we've got a lot of sodiums out here and not very many sodium ions in there. So there's also a concentration gradient. So a bunch of those sodiums will rush in. Now, not all of these sodiums rush in, but enough of the sodiums rush in to actually raise the voltage up to about plus 40. About plus 40. And at that particular voltage, an interesting phenomenon happens. The cell membrane becomes impermeable to sodium, but some specialized diffusion channels automatically open up for potassium. We're at plus 40 millivolts. Potassium can diffuse out now through the cell membrane, and it certainly will. Why? Well, there's a positive charge on potassium, and there's a positive charge in the cytoplasm, so there is an electrical gradient. And there's a concentration gradient as well, because there's a lot of potassium in the cytoplasm and not very many potassium ions by comparison in the extracellular fluid. So we first have an inrush of sodium, and then we have an outrush of potassium, and that's all happening right down here in this one location. When it happens right there, it actually makes a change in the cell membrane right next to it that initiates the same chain of events there, and then here, and then here, and then here, and it moves down the cell membrane at an extremely rapid rate. We call the the movement of sodium and potassium, the raising of the voltage and the lowering of the voltage due to the diffusion of sodium and potassium, we call that an action potential. And as that action potential actually progresses down the cell membrane of the neuron, the moving action potential constitutes what we call a nervous impulse. So basically that is what is going on from a ion movement in a nervous impulse. An inrush of sodium first, and then an outrush of potassium. The voltage goes up, and then the voltage comes back down. Physiologists refer to this rise in voltage as depolarization, and then when the voltage comes back down due to the diffusion out of the potassium ions, they call that repolarization. So we have a very quick and momentary difference in the charge that occurs. And as that scoots down the cell membrane, that moving phenomenon is called a nervous impulse. Any questions about that before we go on? Yeah, Adam?
So how does it reset back to the resting state? Now it seems that the potassium is going to be outside of the neuron and the sodium is going to be inside the neuron at this point. How do we get back?
Good question. I want to go back to the overhead for just a second, if we can go back to the overhead camera. I want to emphasize something. I've just drawn a few sodium ions. Imagine lots and lots of sodium, thousands and thousands of sodium ions out here, and the same with the potassium ions in there. The total amount of sodium and potassium that make this movement that we're talking about is relatively small compared to the total amount of those ions that are present. And so this thing can actually occur many times before we have a significant net movement of sodium in or net movement of potassium out as far as the total concentrations are concerned. But maybe your question is, how did we get this situation in the beginning? How do we end up with a lot of sodiums out here in a resting neuron and a lot of potassiums in the cytoplasm in a resting neuron? And that has to do with what we call the sodium-potassium pump. I'm going to talk about that in a little bit, but I want to wait before we get there. Let's take a look at a graphic that will summarize the things we talked about in a resting neuron. We said that the sodium was low in the cytoplasm but high in the extracellular fluid. Potassium was just the opposite, high in the cytoplasm but low in the extracellular fluid. The cell membrane is relatively impermeable to simple diffusion of sodium and potassium in the resting state, and the cytoplasm is negatively charged, about -70, due primarily to protein ions. Now that's the neuron at rest. Then we'll go to the next graphic and summarize what we said that would happen when we have one of these action potentials. Sodium rushes into the cytoplasm initially, because there are diffusion channels that open up in the cell membrane for that ion, and then those diffusion channels close, but others open up that allow potassium to rush out. When we say rush in, rush out, we're talking about simple diffusion. Simple diffusion, okay? And so we have a rise in voltage, and then we have a drop in voltage. Remember, the rise in voltage was called depolarization, and the drop in voltage was called what? Repolarization. And that drop comes back down enough to, quote, reset it. It gets back down to, actually it overshoots just a little bit. It gets back, for our purposes, gets back down to the resting potential, so it's ready to go again. Bryce?
How long does that take? Because, like, if you grab something hot and hold on to it, it keeps hurting. It doesn't start and stop and start and stop.
Impulses continue to be generated by many of the sensory receptors as long as the stimulus is present, Bryce. So next time that happens, just go ahead and turn loose of it, you know, rather than hanging on. You don't have to say, oh my, that hurts, I wish it didn't. But it continues, impulses continue to be generated, and they continue, those impulses then are carried on into the brain and spinal cord by sensory neurons in many cases, as long as the stimulus is present.
So does it repolarize between?
Oh yeah,
there,
it has to reset, and so that's going to be happening. Of course, it can be different sensory. We've got thousands and thousands. It's an astronomical figure of how many sensory receptors are in a square millimeter of skin, for example. And sensory receptors respond to different stimuli, too. There's lots of different kinds. Because remember, I mentioned a while ago, sensory receptors are actually very stimulus specific, and there's different ones that respond to this stimulus versus this other stimulus. When a nervous impulse reaches the end of an axonic process of a neuron, the junction of that axon with the next cell is called a, anybody know? Eva?
A synapse?
A synapse. Now there's only 3 kinds of cells that a neuron can synapse with. It's going to synapse with another neuron, with a muscle cell, or with a glandular epithelial cell. And for a particular neuron, it only does one of those 3 possibilities. Let's take a look at a graphic which summarizes synaptic junctions a bit. We got another cell, another neuron rather, a muscle cell or a glandular epithelial cell. Those are the possibilities. And what is actually happening at the synapse is not an electrical spark jumping across the gap, but there's actually neurotransmitter chemicals that are released to interact with the cell membrane of the next cell. If we could go to the overhead camera for just a minute, I've got a model here. We can just take a look at this one that actually shows the end of an axon. So main part of the cell would be back this direction, but here's the end of the axon, and here it would be coming up against the cell membrane of a next cell, whatever cell that would be. And these little white beads represent little vesicles of neurotransmitter. There's actually several different kinds of neurotransmitters that are found in different neurons within the body. And when the axon receives the impulse down to this end, some of these vesicles of neurotransmitter are released. They diffuse across the very narrow gap and physically and chemically then interact with the cell membrane of this next cell. And they basically may raise the voltage in that next cell, which might stimulate impulse to occur in the next cell, or they might lower the voltage in the next cell, which would tend to cause an impulse not to happen or prevent the impulse from occurring in the next cell. Let's take a look at the next graphic, which will summarize what we mean by these chemicals. Neurotransmitters are called excitatory if they raise the voltage in the next cell, and they're called inhibitory if they lower the voltage in the next cell. So some of them raise, some of them lower. And these are just big classification categories. There are many individual excitatory neurotransmitters and several inhibitory neurotransmitters as well. Adam?
So does each axon contain both types of neurotransmitters?
No. Typically, we think of one neuron having only one kind of neurotransmitter in it because the neuron wouldn't be in a position to make a decision in and of itself as to which neurotransmitter to release. So, yeah, Bryce?
So like with painkillers, do those just stop the transmission by putting chemicals in the way, or?
Well, there's a lot of different ways that painkillers work. Some of them work peripherally and some of them work centrally. So some of them work actually on the brain and some of them work out in the peripheral nervous system. Some of them inhibit nervous impulse transmission. Some of them change the sensitivity of pain receptors so that the receptor doesn't pay as much attention to the stimulus as it might normally under different circumstances. So a lot of different ways that painkillers work, depending on the particular one. One of the things that we didn't talk about today, and that I want to spend just a little bit of time on, is, and we'll go to the overhead camera for just a minute, I want to go back to what we talked about on this cell membrane difference in concentration of sodium and potassium. See if I can get this line a little bit darker here. So we said that there was a lot of potassium. Here's the nucleus of that neuron. A lot of potassium inside this cell membrane and a lot of sodium on the outside when the neuron is at rest. And we never did establish how that came to be. There's actually an active transport mechanism that's located all over the cell membrane of neurons and most other body cells, in fact. It's called the sodium-potassium pump. And we'll just write it like this, sodium-potassium pump. And this pump actually moves sodium out of the cytoplasm and potassium in. So because it's moving the sodium out, it will pump the sodium ions out to the extracellular fluid, and it also moves potassium ions in to the cytoplasm. And so the reason I want to bring this up, this is how we have the original difference in sodium potassium ion concentrations. It's due to this active transport mechanism called the sodium potassium pump. Now, like other active transport mechanisms in the body, it's powered by ATP. And physiologists have worked out that for each ATP expended by one of these pumps, and a given neuron will have thousands of them over its cell membrane, but for each ATP expended, we move 3 sodiums and 2 potassiums. Of course, those 3 sodiums are going out, and those 2 potassiums are going in. So 3 sodiums, 2 potassiums. 3 sodiums out, 2 potassiums in for each ATP expended. Now these pumps work 24 hours a day, 7 days a week, 365 days a year, and they have nothing to do with the impulse transmission while it's occurring. But they're absolutely necessary to establish the difference in sodium and potassium concentrations necessary for an impulse to occur. It's kind of like when I set a mouse trap and put that mouse trap underneath my kitchen sink. Then I go to work. Now, the mouse has his free agency as to whether he's going to mess with that trap or not. So I don't consider myself directly responsible for his demise if he gets caught, you see. But I had to set the trap in order for him to be caught. And the same thing here. The sodium-potassium pump is not actively involved in the nervous impulse transmission at the moment that it occurs, but the sodium-potassium pump is absolutely necessary to establish the difference in sodium and potassium concentrations that are required for the impulse. Does that make sense? Mm-hmm. So it's a kind of an indirect relationship, and so I just want to make sure you understand that the pumps are working all the time. Matter of fact, most body cells have these. It's said that a very large percentage of the total ATP, the energy that's expended internally by our body, is actually used to power sodium-potassium pumps. They're very critical to the function of neurons and muscle cells, but as I mentioned a moment ago, most body cells have sodium-potassium pumps within them. And so these things are they're working all the time. If we poison these sodium-potassium pumps, we'll die very quickly, in fact. You have any questions about the sodium-potassium pump or anything else we've talked about today? Eva?
What exactly is it that does the pumping? What is the pump made of?
Well, let's go back to, it's a concept. It's a physiologic concept. Let's go to the overhead camera for a minute, and let's just go back to what we said before in regard to active transport. Active transport is a way to move chemicals across membranes, but it requires what? 2 things. We have to have a carrier. Usually those carriers are some kind of protein in most cases. And for active transport, we also have to have energy in the form of ATP. So what we're saying is we've got an active transport mechanism. We know what it moves. It moves sodium and potassium. And so we're going to call that, because it moves those across the cell membrane, one direction, one the other, we're going to call it the sodium-potassium pump. So it's, I don't know exactly how it's structured at the microchemical level, certainly. But these, these items we've got down here conceptually are what we need to know about it. Again, sodium out and potassium in. Powered by ATP, there has to be a carrier involved to move these ions. In other words, they don't freely diffuse, because we're moving them against a concentration gradient, aren't we? In the resting cell, in a resting neuron, the sodium is continually pumped out, even though there's a large concentration of sodium already out there. And the potassium is pumped in, even though there's a large concentration of potassium already in there. Now, we said they're working 24 hours a day, 7 days a week and all that. Obviously, if they worked continually, eventually, in theory, we would get all the sodium pumped out and all the potassium pumped in. What actually happens is, once we reach a certain level of sodium concentration outside the cell membrane and a certain level of potassium concentration inside the cytoplasm, actually, when we get the concentration to a certain level, then sodium and potassium will diffuse to a limited degree back the other direction. It's kind of like you can only put so much water in a glass until it overflows. Well, the sodium-potassium pump can only move so many sodiums out and so many potassiums in until some of them start diffusing back through the membrane, even though the membrane itself is not very permeable to that diffusion. It takes a very high concentration gradient for simple diffusion to occur in a resting neuron until we reach the threshold level in which the whole series of events kicks in automatically. Any other questions in regard to nervous impulse transmission or neurons or glial cells, anything that we've talked about today? In our next session, we're going to be talking about the peripheral nervous system. And again, PNS, remember from what we said at the beginning of today, includes nerves, ganglia, and sensory receptors. So that'll be the thing that we'll concentrate on at that time. Sometimes you'll hear people say, well, there's 4 things in the peripheral nervous system: cranial nerves, spinal nerves, ganglia, and sensory receptors. And certainly we wouldn't take issue with that. The nerves can be divided into those 2 categories.